Now we're good. All right. Thanks everyone for joining. I'm excited to tell you about some work we've been doing over the past, I think this is like a 2 or 3 year project. Um, I'll have to say just kind of at the outset that I have a general interest in aquatic plants, and obviously working with something as exciting as bladderworts has been really fun for me. I would say I'm not an expert per se on bladderworts. I haven't spent a ton of time studying them, but this whole project kind of came about as what I do a lot now for research is basically try to understand diversity, do a lot of plant identification using DNA. So that's kind of my connection is I have some contacts in different parts of the world and the country that can send me plants, and that sometimes turns into the start of a fun project. So I'll explain how this got started, and hopefully some interesting results along the way. Okay, so as carnivorous plant lovers, I'm sure you are well familiar with this group. A little bit, maybe not one you run into in your average field exercise. It's typically an aquatic plant, at least the ones around here primarily are. I'm in North America. And kind of noteworthy as an underwater carnivorous plant. There are not many of those, and has some rather bizarre modifications. So even the things that we call leaves, the jury's a little bit out as to whether we should call those leaves or stems. Typically, you don't see leaves with structures, rather complex structures coming off of them like we do here. So whatever these are, leaves or stems, a lot of these bladderwort plants have their characteristic bladders, which shown here in a microscopic image is just a little pouch that has trigger hairs on the end, and in the presence of a food item, there's some trigger hairs on the sort of the door of this trap, and it sucks the animal in through vacuum. So it creates a space that wasn't there before, and in the process pulls the water and the food inside of its traps. And so the plant has tons of these little traps all throughout, and this is how it is carnivorous. Typically, as you're also probably aware with carnivorous plants, these are plants that grow and have a bit of a competitive advantage in habitats where nutrients are poor. So if they're in a low nitrogen, low phosphorus situation, they can supplement their nutrients by ingesting animals that have those elements that they need. So I should also maybe early on express some regret that I have not credited many of these images. So I'm very grateful for the images I was able to find online, but most of them, I'd say maybe all of the plant images are actually not mine. I would love to credit them. I kind of ran out of time on that. Okay, so why do we need to study them from a diversity standpoint? Well, there's certainly a lot of diversity among species. There are about 240 species of bladderworts, and they don't always have those characteristic bladders. As I showed you, sometimes they're rather richly present on the, I'll just go ahead and say leaves, as in this common bladderwort, Utricularia macrorhiza. Sometimes they're a little harder to find, the bladders, species like Utricularia minor, intermedia, and so on. Some of them are free-floating plants in aquatic habitats. A lot of them are kind of bog plants that are growing out of wet soil, and the soil is maybe very organic, so they have some sort of spaces to trap animals or under the water surface, but not necessarily free floating in the water. A little bit of diversity in flower color here is showing, and then a handful of them have these interesting, basically air tissues that certain leaves are modified to support the flowers above water. So definitely interesting and worth studying, I think. So kind of at a getting us to the context of my study, this all started, and a lot of my projects start as people encounter plants and have difficulty identifying them. So identifying plants is a matter of being well-versed as a botanist. And then depending on the plant groups you're studying, you might have to be a bit of an expert in the group that you're studying for you to confidently identify them. And the bladderworts are a little bit extra difficult, I would say. They're in this aquatic plants category, which typically a lot of botanists kind of shy away from aquatic plants because often they don't have reproductive parts. Often they have modified tissues that are a little harder to make sense of. And often they're in habitats that are not typically what people are running across. So this is a key from the Flora of the Southeastern United States that came out recently, a revised edition of that, giving you a sample of species you could find in the United States. As we move forward in time, the ability to identify plants, we're getting new technologies that can help us with that. And I teach classes on plant identification, and it's always an interesting process to kind of watch this landscape shift as we go. Where, originally there was only the book, sometimes some illustrations, and now there's this widespread presence of artificial intelligence and other systems that can identify plants for you. You take a picture and you get an identification. So it's kind of the new way of things. We'll see how it settles in the context of the old way of identifying plants. As I mentioned, underwater plants sometimes not having their reproductive parts and therefore a little more difficult to identify. And so this might leave you, even if you have a flora guide, you might leave you unable to identify a plant confidently if you're missing flower or fruit parts. A lot of times those are key elements for identifying a plant. So what to do? Well, one option that's also in the technology realm is to do DNA sequencing. Just like artificial intelligence and photography-based identification, this is a technology that's getting easier, faster, cheaper as time moves on. And so this is basically the realm I occupy now in my own research, where interesting plant identification questions come up, and we take the DNA approach. So an example here of a plant and its DNA sequence. This is just an illustration of the sequence as we get it back. It looks like the letters are called out. These are the 4 nucleotides of DNA, A, C, G, or T. And the order that they appear is characteristic of whatever gene you're looking at, whatever organism you're looking at. And you can often match those, and that's a way to do your identification. So plants, just another throwback to some intro biology stuff. Plants have DNA in a variety of places. They have a nucleus, just like we have in our cells. They have mitochondrion, like we have in our cells. Both of those have DNA, genes and DNA that we could sequence. And then plants also have chloroplasts, which have their own DNA. And depending on what answer you want to get, you might look for DNA in different parts of the plant. Typically, people are using chloroplast DNA. It is reliable for getting that kind of DNA. It's abundant in cells, so you can have multiple chloroplasts per cell. And for better or for worse, the chloroplast DNA, you typically get one kind per cell. The nuclear DNA, the DNA from the nucleus, sometimes you get 2 copies of the same gene in the same cell. And so you can have, if you go back to your genetics classes, right, you have 2 alleles for the same gene. Sometimes that's a problem. And then there also can be problems if plants hybridize or have multiple copies of their chromosomes. You can have a little more trouble with nuclear DNA. So nuclear DNA, potentially a problem, potentially also, as I'll show you, is one way that we get some answers about interesting plant questions. All right, I'm just going to show you this really quickly. This is just an example of how you can go from the plant material itself to the extracted DNA. We're basically purifying the DNA molecule so that we can work with it. And then there's more DNA sequence. Once you get your DNA sequence, the job is to align sequences among every plant that you've studied. And this illustration shows you basically when you have an alignment, you want to have a certain amount of these nucleotides that are almost constant or absolutely constant. This gives you a manner of confidence in your identification, or sorry, in your alignment. So every one of these columns we're sort of implicitly comparing the letters. So if everybody has a T, that's not super informative for diversity, but it is informative to tell us that we're pretty sure we have the same region. So this left-hand area, everybody's almost identical in that area. That tells us, yeah, we probably sequenced the right gene. This middle area, there's some more variation. So whether in spacing or the letters of any column, all those differences, if there are plants that have something in common, then that's taken as sort of one of many pieces of data that say that they're closely related. And so we throw this at a computer, and the computer tells us which ones of these are most closely related, and then sort of next most closely related, and next, and so on. And we end up with a phylogenetic tree, which I'll show you some of. All right, so here's an example of a phylogeny. This is of duckweeds. This is another project I worked on. And the duckweeds are kind of a pet group of organisms for me as well. And a nice part about those is that there are pretty extensive DNA sequences for all the species of duckweeds. So I can say comprehensively that we pretty well understand the world's diversity of duckweeds. And so you can use these sequences that have been published before by someone else and use that as a basis for identifying a new organism that is unknown. So if you get a duckweed of any kind, you should be able to sequence one of these target genes that's already been sequenced and get an exact or almost exact match to what is already known. So this is what we call DNA barcoding, kind of like a supermarket barcode, right? We're matching the signal to the database of what we know, and that's one way of identification. Some people lose a lot of sleep over correctness of barcodes, but I think you can kind of ballpark it pretty well. So this is kind of a modern sensibility of getting as much sequence data as we can possibly get. There are people spearheading projects where they're just identifying all the plants they can get their hands on. There's like a flora of Canada and flora of Florida, I think, is doing one. Wisconsin had one recently where they basically had representation from every species. And so if we have this background database, then any new unknown plant can be identified. Not only that, but even like fragments of a plant. If you had a leaf, if you had conceivably a grain of pollen, you'd be able to get this kind of data from. So real neat potential for moving forward in the answers we want from plants. Another example from another project I was working on. This is also kind of illustrating this identification through barcoding. This was a daughter plant, Cuscuta, and we published a paper on this plant that was found on an aquatic plant in Texas. So it was actually growing underwater, not a usual place to find daughter. And this is another kind of plant that doesn't have a lot of features that you could use to identify it. So we did the DNA, and we found, just kind of highlighting here, the sequences that we got were exact matches to a known species. And so we used that as evidence to identify the unknown plant. All right, so that's kind of my, we kind of got off track from bladderworts for a little while. Kind of context about my research interests and the progress I've made on helping people to identify plants. All right, so coming back around to the bladderworts. I also am hardly the first person to do this kind of work on bladderworts. So even before our project started, there was a fantastic database of sequences for species, and of those about 240 species, 78 of those species were represented in this study, and that helped to kind of clarify some relationships, some taxonomy. So, which category do you put your species into? These are taxonomic subgenera. So over here, subgenera, and then the nearer label are taxonomic sections. These are all ways to categorize and make sense of this big diversity of bladderworts. So this was a tree that was available before my work. And then this is a little bit more zoomed in. I'm going to sort of make the case for why new work needed to be done. So the study there from 2018 had great representation, but was surprisingly, I would say, a little bit lacking in North American plants, which is totally fine. It was published by authors in South America. They're going to sequence their plants, and then I guess we can sequence our plants. One of the things that stood out to me, so kind of the origin of this whole big project, really came out from a couple of species here. These are North American plants that both have this inflated support system that keeps their flowers above water. Very obviously, I mean, a lot of bladderworts can still support their flowers out of water, but this is a really contrived version of that, where they have this spongy parenchyma tissue in the support structures. So one species called Utricularia radiata, and another called Utricularia inflata, radiata, inflata, however you want to pronounce those. They are very similar plants. And looking at the phylogeny, they don't come out as super closely related. This shows some uncertainty basically in the relationships, but I kind of would have expected them to be just sort of buckled up right next to each other as very closely related plants. And digging a little more deeply, there's this underlying problem where the existing data, even though there's extensive data, the existing data were not equivalent for these two species. And so I probably should have clarified here, the lightly colored boxes are where data were actually published for those species. And then the dark ones are where data were missing. And so if you compare Utricularia inflata and Utricularia radiata, for every one of these gene regions that was being studied, it was present for either one or the other. So you could sort of put them in the phylogeny in context by finding another closely related species, but they could not, there was never a chance for them to be very similar to each other because we didn't have the data from the same genes. So it kind of explains why they're not coming out as closely related. It also highlighted the fact that if you wanted to try identifying a plant, these are the North American species. If you wanted to identify one of these, there's some gaps where if you sequenced a particular gene region, you might come up empty for a species. So these are, I'm not going to dwell on the details of these, but they're different gene regions, 4 from the chloroplast, 1 from the nucleus, and not every species is represented for any of these. So we don't even have a single one of these could be used as that so-called barcode. Okay, so a few questions. I'm just going to sort of raise questions and answer questions as we're things that came out from this study. One of the questions for, I guess, sort of a question for North American plants, but also a global question. Our most common bladderwort is this Utricularia macrorhiza, the common bladderwort. We were talking before the presentation that this is, most likely you're going to run into this species. And there's been some uncertainty about whether the common bladderwort in North America should be considered as its own species, Utricularia macrorhiza, or if it should be a subspecies of Utricularia vulgaris, this more European species. And I'm showing you a map here from Plants of the World Online. That's a resource I really enjoy for giving a updated version of where species are native. And so I took their maps and I color-coded them a little bit differently so you can see the contrast. And this maroon color, the reddish color, purple, is Utricularia macrorhiza, North America, and Eastern Eurasia. And then the brownish color is Utricularia vulgaris. So this is basically as they're defined through Plants of the World Online. And it's my preference, I'll just say it's a preference, to treat them as separate species. And I've listed on here some other sources that might still maintain them as separate species. So that's one of the beauties of taxonomy is we can still disagree about things. Okay, so within that question, here are the species that I was talking about, Utricularia vulgaris, Utricularia macrorhiza. They are part of a rather widespread and diverse group of closely related species. And so I've highlighted it on the phylogeny. They're all very closely related. Sometimes you'll encounter a DNA sequence that's not really different from one of these species to the next. And so it kind of makes sense that there's some uncertainty as to how to classify these. Sometimes if they're so closely related, then you might want to have something like a subspecies designation for them. So Utricularia vulgaris, macrorhiza. There's another species, Utricularia tenuicaulis, which is also a northern hemisphere species. And then I'll show you some papers that are sort of outlining these as hybrid species. So Utricularia japonica has been identified as a hybrid between macrorhiza and tenuicaulis, and Utricularia neglecta has been identified as a hybrid between tenuicaulis and vulgaris. As you're identifying these, I'll just point you to this paper if you wanted to look. They've got a nice table to highlight the differences between the species. And a couple you can see on these images are their corolla lip. So tenuicaulis in particular has this kind of flat corolla lip. Utricularia macrorhiza, more deflexed. And then macrorhiza has this rather long nectar spur. If you catch it in a photograph, you can much more easily see the nectar spur from the side as you're looking at Utricularia macrorhiza versus these other species. So again, maybe some good evidence for keeping these as separate species besides just the DNA evidence. Besides the ones I had on the previous slide, there's also a couple. Utricularia stellaris, Utricularia australis also fall into that closely related group. Because they're closely related, I think that's where the taxonomic uncertainty comes from, is that they are very similar morphologically. But if we find reliable DNA markers to separate them, that can be useful. So this, from the same study as the flower images I showed you a couple slides ago, this is a study that centered in Eurasia and was highlighting basically the boundaries of Utricularia tenuicaulis, Utricularia macrorhiza, vulgaris. And this study provided some of the best evidence to date of the fact that Utricularia australis, this one that I've got here is a map for Australia or Australasia. They put forward the idea that this was just an Australasian species. And even though a lot of plants in the Northern Hemisphere were being called Utricularia australis, a lot of them turned out to be one of these hybrids that I showed you before. Okay, so the evidence of that is from the DNA sequences as well. A different study, also from Eurasia, was highlighting a different kind of hybrid species called Utricularia ochroluca, is one that they have in Europe. We also have that, or plants that go by that name in North America. And they're proposing that this is a hybrid. They sequence some individuals, and sometimes they come out as closely related to Utricularia intermedia. Sometimes they come out as closely related to Utricularia minor. This yellow one here, Utricularia stygia, was basically wrapped in with Utricularia ochroluca. So these are in a different group of species, but also showing some sort of uncertain relationships among the individuals. So they took this is one individual that matches Utricularia intermedia. This is one individual that matches Utricularia minor. They take that as evidence for this species being a hybrid. It doesn't quite conform to the, I'll say, like more of a sure thing conformation of hybrids. So this is a different group of plants, the water milfoils, showing the kind of evidence we often use to confirm a hybrid, where you'll sequence a gene and get two signals at the same place. So both of these that I've highlighted have signals. The black is G and the green is A. They've got both signals present in the same place, and the background is everybody is otherwise the same. And so this is evidence where the one kind of sequence matches Myriophyllum heterophyllum, and one of them matches Myriophyllum laxum. And this is from the ITS gene, which is from the nucleus. So this is one of the advantages of sequencing nuclear genes, is that you can have these different versions And these different versions of DNA are a really good way to confirm a hybrid. So we didn't have exactly that kind of confirmation with the Utricularia ochroluca. And their study was in Europe. And so part of our study, we were able to get some material from Utricularia ochroluca in the United States. And it's not a widespread species. It's got a kind of odd distribution map. It's sort of a one-off in a variety of places. We got our material from the Pacific Northwest, from Oregon. But otherwise, it's kind of sprinkled rather haphazardly throughout North America. And our evidence was consistent with what was reported in Europe, but also potentially what we're seeing here is that it's not any different from Utricularia intermedia. So again, if we were looking at a hybrid, we might see some DNA that matches the other supposed parent, Utricularia minor. Everything we sequenced was just a match to Utricularia intermedia. And again, this is a taxon that's not super differentiated morphologically from that other species. So potentially they're the same, but as a rare plant, this is one that has kind of special status where it grows. So I'm not going to make a strong claim about making it a synonym, but I'd say maybe we need more study or something like that. Okay, so not definitively a hybrid as proposed, at least in my opinion. Okay, so this is our total phylogeny at this point. Again, almost everything on here is someone else's data, so I can't take credit for the bigness of this tree. But it does have highlighting for the species you can find in North America, and they're scattered throughout. So this ends up being an interesting pattern for the North American bladderworts, is that they don't have a single common origin. They don't even have, I don't think they even really have a small number of origins. It's rather quite a few. 7 of plants that, so these colors go along with the map. And the pie chart at the branches leading to these North American species tell you where we have reconstructed the ancestor to be. So if you take the green North American plants, almost always their ancestor is reconstructed to be this yellow neotropical version. So they're related to plants from Central and South America, and it looks like that's where their ancestors were, and they independently dispersed 1, 2, 3, 4, 5, 6, 7 times. If you think about natural dispersal over long evolutionary time, this is maybe one of the more conceivable ways that plants could disperse. They're not crossing any oceans to get from South America to North America. And so it totally makes sense. There's some migratory flyways for birds and so on. We do have, though, those species that I mentioned are closer related. They're up in this kind of tight group here, where the green are North America, and they're scattered among a lot of these Eurasian plants. And so a small number of the North American plants, the Utricularia macrorhiza, the common bladderwort, also intermedia, minor, ochroluca, those are reconstructed to have an ancestor potentially into North America and then dispersed into Eurasia is the strongest evidence right now, actually. And then so this group has kind of a few dispersals to or from North America, Eurasia. Okay, so a few more kind of one-off mentions as we're going through. So kind of coming back around to the inflated bladderwort, Utricularia inflata. This one of the reasons we wanted to identify it at the start of this study was it's showing up in much more abundance than historically was present. So these are photographs from Connecticut, and Connecticut on this map is up in the farthest extent of the range where it's claimed to be not an abundant plant. So it's surprising to see a plant that has been in the area for a while suddenly taking a really aggressive growth form. And so one of the questions was, is it really this plant that we think it is? And so we confirmed, yes, definitely, these are Utricularia inflata plants. And I guess it'll be up to the ecologist to find out, why this plant is doing so well. But this is a pattern we've observed in a few species where they become problematic at the northern ends of their range. So potentially this is a result of climate change where winters are warmer, summers are hotter, and these are favoring kind of a geographic shift in plants. Just highlighting here some notes with the dates on there for people making notes about these inflated bladderwort plants showing up in places where they weren't known to be super abundant. New York, Massachusetts, and then this one is Washington State. All right, so it's actually kind of a small number of species that are in this group of plants that have the inflated flower support. So just put them on the map here. Radiata inflata are North American. Then we have Utricularia platensis and breviscapa that are South American. There was a report, along with some DNA sequences, of Utricularia platensis showing up in Japan. We included that data as part of our study. So down here, Utricularia platensis. This is a phylogeny that shows the all the sequences that we had for the chloroplast genes. And what it's highlighting is just a clean differentiation between the clade, this group of sequences that are Utricularia inflata at the top, are clearly differentiated from this group that are Utricularia radiata at the bottom. And we aren't quite sure if this should continue to be called Utricularia platensis. It's coming in as a very similar species to our Utricularia radiata. I guess we'll see. Maybe this could be cleared up over time. Okay, along with our project, one of the co-authors is a person who works in Ohio, and this one highlights the advantage of just like surveying, kind of staying up on identifying what's around us. And so a lot of my co-authors end up being people who are familiar with plants in the field, and they will notice something unusual and send it to me. These ones didn't look unusual at the beginning, but they ended up having a rather unusual genetic outcome. So we would expect of these species, we've only expected the Utricularia macrorhiza to be present in North America. And two places in Ohio, we found one group of locations had this hybrid Utricularia neglecta, and then a separate place had Utricularia tenuicalis. Both of these are not known to grow in North America before our study. I don't know if anybody would have caught on to that. It's the kind of plant that, if it's not on your radar for your local flora, you would not suspect. It's convenient when you're identifying plants to work with a small number of expected species, not 240 possible global bladderwort species. But sure enough, this is the DNA sequencing for these plants, the purple ones on the map there. And if you take the sequences and separate them out into individual sequences, one of those matches Utricularia tenuicalis. One of those matches Utricularia vulgaris, which also not present in North America. So whether that was a hybrid that was brought in, or whether some sort of hybridization happened in North America, all of that is possible. With water plants, there's often introductions of new species from people's water gardens. So I know we have some plant collectors as part of our audience here, just to reinforce the idea that it's important to prevent escaping into wild habitats that could have been the source for these. Certainly, this is the only place we know of them existing right now. And we'll try to keep from having any new infestations that cause people problems. Okay, so kind of wrapping up here. All of this kind of started because I have this service that I provide where people can send me plant material and I identify it. A lot of times that turns into a full scientific project. I feel like it's valuable to publish the results, but my day-to-day, especially during the summer season, a lot of that is spent just sort of helping people put the correct names on their plants. So some examples of plants I've worked with, this Japanese knotweed on the left, hydrilla plant in the middle, and then just another example of putting a correct identification on something from Nymphoides project we had. So thank you all for your attention. I have a lot of people who helped out with these projects, and very grateful for their help sending samples and helping with the analysis and writing. And we'll see where the next project takes us. So thanks for your attention. I'm happy to take some questions.
Okay, thank you, Nick. All right, so I'm going to talk briefly about the ICPS. And then you have a couple of questions that are in the chat, which I will read in a moment. And if anyone else has questions or comments, they can type them into the chat now. And thank you everyone for attending this live webinar. So my name is Kenny Coogan. I'm the Education Director for the ICPS. And the ICPS is about 1,300 paid members who receive a beautiful quarterly full-colored newsletter. We offer free webinars to the public. Here are a couple of the webinars that are coming up. We're having kind of 2 a month for the next couple of months. And Nick's presentation was great. It was a little scientific. Some of our presentations are super scientific. Others are about growing. Some of them are funny. And you guys can RSVP to all of these. And if you can't attend them live, because we are international and they're at different times of the day and different days of the week, all of our webinars are posted on our YouTube page. If you're interested in presenting a webinar, you can send me an email, which is down below. And in the past, we've had people who own carnivorous plant nurseries, scientists, growers. If you go to our website, carnivorousplants.org, you will see that we have 5 different animated videos for beginner carnivorous plant enthusiasts. And all of these come with worksheets for kids in the classroom. We encourage you to utilize these free resources. And then we have dozens and dozens of scientific webinars that you can also watch for free. The first Wednesday of May every year is World Carnivorous Plant Day. And if you go to our YouTube channel, you will see that we have about 20 videos for every year for World Carnivorous Plant Day. And you can be entertained and educated about carnivorous plants for hours and hours and hours. If you would like to become a member of the ICPS, that would help us keep things running. You can become a regular member where you receive that beautiful newsletter that I was mentioning, or you can become an e-member. And both types of memberships, you get access to all of the carnivorous plant newsletters, which span 30 years. So that's a lot of research, a lot of information. And in those newsletters, we have scientific papers published, as well as cultivars and some field trip reports. And a lot of people who are growing carnivorous plants like to know when a cultivar was named and published, what are the characteristics of it, who made it, and all of that is available on our website. To go to our free website, it's carnivorousplants.org, but to get access to all of our carnivorous plant newsletters, this is the website right here, icps.clubexpress. When you become a ICPS member, you get access to our seed bank. We just did an update maybe a week ago. The seeds are very inexpensive. We can ship to many countries. And to learn more, you can contact our seed bank manager. In August every year, we have the Carnivores in the Classroom grant, where we fund about 40 teachers around the world with $150 worth of carnivorous plants to the classroom. So this is where we get nurseries to donate plants, and they ship them directly to classrooms. And then teachers can implement using carnivorous plants in all of their curriculum. What's really cool is that in order to receive that grant, teachers have to submit a lesson, and all of the lessons are available for free on our main website, carnivorousplants.org. They're separated into K through 5, middle school, and then high school, and you guys can check those out. A lot of them come with a PowerPoint and a worksheet, a PowerPoint and a Word document, PowerPoint and a Google Sheet, and we encourage you to use those resources as well. This year, these are the nurseries that funded all of our teachers. And in the middle, we have Carnivorous, which is a 3D printing company, and they make carnivorous plants. And they make them out of sustainable, recycled, different materials. And very soon, we will have the instructions for these 5 models available on our website for free. So if you have access to a 3D printer, you can choose whichever one you want and you can print it yourself. And as Nick was talking, I added on the slide our Utricularia inflata. And we have a store, it ships internationally. We have Aldrovanda, Utricularia inflata. We have a Pinguicula, Cephalotus, Drosera capensis, Nepenthes bicalcrata. And this ships internationally. Proceeds benefit our education and our conservation funds. You can get our logo with these different types of plants on tote bags, mugs, tank tops, t-shirts, sweaters, hoodies, lots of different things. And every other year, we have an international conference. A couple of months ago, we were in Vienna, Austria. The previous time we were in Japan. And in 2026, we will be having an ICPS conference in the Americas. So that could be North America, Central America, or South America. We are going through the bids right now, and we will keep you updated. All right, thank you, Nick, for that commercial. Okay, so the first question, Nick, is, okay, we understand that you're looking at the sequence of a specific gene and you're looking at the base pairs, which could be one of 4 things, A, T, C, G. And how do you account for genetic diversity within a species? Because I'm thinking of humans, we have very similar DNA, but there's a lot of genetic differences. And how many, if you get a sample, do you need 5 of the same plant or 100 of the same plant to know what's what?
Yeah, great question. So a few of my slides showed multiple individuals of the same species. So I guess in my experience, as I've done this a few times, there's often very little variability. I mean, it gets into really big questions, right? With the origin of a species and how big is the range, and are individuals mating with each other across the entire range, and so on. So there are some people who they devote their scientific career to asking that question of, is it cleanly one species different from another? Mostly, like I said, in my experience, it tends to be pretty clean, that if you have named species, very often they have, there might be some variation within each species, but the differences between them are consistent. So it's always something to keep in mind. And certainly if we had one individual of every species, I would have less confidence in making some of the statements. But generally, yeah, this one, I'm showing you right now is a lot of sampling, probably more than we had to. But a lot of this, I think, came out of maybe Connecticut, where they were trying to get clarification on as many plants as they could. So sometimes I get a lot of samples of the same species. Sometimes I get just a couple.
Okay, Mark asks, are the rRNA 18S sequences too broad to use for species differentiation within this genus.
All right. Okay, so I use the gene region called ITS, which is a spacer region. This might be something of interest to some of your audience. So basically, when you're trying to build a phylogeny, you want DNA that is variable. You need those differences so that you can highlight, one species different from another, and also similarities that, all individuals of the same species are almost the same. The ITS region is known to be very variable. So almost always there's some difference between named species. And the ITS region that's been mentioned is much less variable. So it's a structural RNA gene, and it really can't change much over evolutionary time. So the differences arise as a result of as a result of mutations, and regions like spacer regions can mutate a little more freely without consequence to the organism surviving. So the ITS is much more slow. But I don't have any data specifically to the ITS region for bladderworts. I would expect it's maybe not variable for big groups of species. So like those species, the common bladderwort and relatives, there might not be any variations. Wildly speculating here. But probably you could get a signal for the whole genus using ITS. So yeah, generally I think I would say yes to that question. Too broad to use for species differentiation would be my short answer.
Okay, Fred asks, is dispersal by bird feet documented?
I don't know.
I did a quick search.
Oh, thank you.
And the only thing I could find was from 1968 for Utricularia olivaceae. And Beal and Quay suggested that waterfowl and shorebirds and herons could transplant fragments of the plants, but I haven't found anything since then. And kind of since then, if you look up Utricularia in birds, they talk about the 2 to 4 Utricularia species that are pollinated by birds. But I mean, for other aquatic plant life or large storms like hurricanes we talk about, that's how small plants and animals could be moved around.
Yeah. Yeah, usually it's the same. I feel like I can conceive of plants moving around. It's easier to conceive if you can put your plant on a migratory pathway. Aquatic plants are in the habitats of migratory birds, waterfowl, and so on. So, and again, to move them around, all it takes is one every 1,000 years, 100 years. So maybe not at a rate that we would easily observe. Fragments on the feet, on the feathers, ingested. I don't know what the seed ingestion is. So sometimes that's a possibility where a water plant will move inside.
So Fred asked specifically by bird feet, and there is research that says that some seeds of Utricularia are dispersed by birds.
Okay, great.
All right, Nick, can you see Fernando's question?
Yes.
Okay, can you read it out loud to make sure we pronounce everything correct?
Okay. He says, I saw a lot of Sanger electropherograms. Have you tried any NGS methods? NGS is next generation sequencing in your work. Whether large-scale barcoding, RAD-seq, or even whole genome sequencing. I did a little bit of the next generation. I'm quite a newcomer to that, and honestly, I feel like the generation is shifting over where somebody younger is going to take that on. So yeah, almost everything is Sanger sequencing, which is technology that's been around since the late 1970s. The newer technology, they call it next generation sequencing, and it's a giant shift in the amount of data you can get. So you can throw small plant particles into your analysis and get millions of base pairs. When I get a sequence, it's like 500 base pairs. So the potential is amazing. You would potentially, with the right time and money, you could get entire genomes for species. I know there's a good handful of chloroplast genomes published for bladderworts. So I guess my answer generally would be I'm going to leave that to other people with better means to get the answers. I have the ability to do a little bit of next generation sequencing. I use that for situations where we have those two versions of the same gene, the polymorphic sequences, to clarify those. But otherwise, I'm still largely in Sanger sequencing.
All right, well, thank you. Thank you, Nic, for doing a great presentation, and thank you everyone who attended this live. And in just 4 tiny days, we're having another webinar. So if you guys can attend that, that'd be great. And this will be posted on YouTube in a couple of days. And thank you once again, Nic.
Thank you, Kenny. Thanks, everyone.
It's not a surprise that gardeners, educators, and scientists are fascinated by these unique plants. The International Carnivorous Plant Society, or ICPS, not only love these plants, but welcomes growers just getting started all the way through professional scientists. The ICPS even started an annual World Carnivorous Plant Day to celebrate them. The free online event is held the first Wednesday of May. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.